So when we're taking a look at generation time and growth and binary fission of our prokaryotic organisms, we have what's called as the growth curve. So when we have microorganisms that are grown in a closed culture, also known as a batch culture, this is a culture in which no nutrients are added and most of the waste is not removed. So when we're talking about a batch culture, what we are often referring to when we're talking about growth curve and batch culture is a petri dish, for example, an agar plate that has some bacteria on it, and that's a closed culture. We're not adding any more nutrients. The nutrients that are in the plate itself are the nutrients that those bacterial cells are going to get. We're not adding any more, and we're not taking away most of the waste. And by most, I mean many organisms will release things like carbon dioxide or oxygen depending on the organism, and some of that is going to be dissipated, for example, in a tube or in a plate, but the other waste products from the bacterial cell are going to stay within that plate and stay sitting there. So we're not taking that out and we're not adding more nutrients. So that would be a batch culture or a closed culture. And when we have organisms in a closed culture, in a batch culture, they follow a reproducible growth pattern, and that's what we call a growth curve. So we see the same thing happen anytime we plate a certain organism. We can take a look at their growth curve and we know exactly where they'll be at certain times of their growth. So an example of a batch culture in nature, then, if we're not talking about a petri dish or an agar plate, then what we could see would be like a pond, for example. If we have a small pond in a yard, and we have a small number of cells that are growing in this closed environment. So let's say we have some bacteria in this pond, or maybe some small little baby pool type thing in the backyard. We're not really adding any more nutrients to it. We're not going to be adding anything else to it to allow those bacteria to grow, but we're also not taking away the wastes in the fluid, in the water, for example. Culture density is another term you should know. It's defined as the number of cells per unit volume, so the density of the culture, how much stuff, how much culture is in a certain amount of fluid. And in a closed environment, the culture density is also a measure of the number of cells in the population. So since we're not adding anything to it or taking anything away, when we're talking about a closed environment, then that culture density, the number of cells per unit volume, is also just going to be the number of cells in the population. So on our agar plate, we're not adding any other organisms to it, so the number of cells that are on that plate would be our cells in the population, which would also be our culture density. So infections of the body don't always follow this growth curve, however, because we don't have a so-called closed culture or batch culture. We, for example, in the human body, will be adding nutrients constantly, right? We're bringing in food, we're bringing in water or other fluids, so we'll be adding nutrients to the body, which would be the petri dish in that case, or the agar plate within the body, or say broth tube, because we're mostly liquid. So we would be adding nutrients, and then we would be taking wastes away. So any waste that the bacteria are producing, we would release in either feces or urine or breath, depending on the type of organism. However, but correlations can exist depending upon the site and the type of infection. So lots of research has been done on the organisms, and depending on the particular bacterium, depending on the site that it's located in, the type of infection, we do have some correlations, some things that we know about how fast things will grow in the different stages they'll be in at certain times. So when the number of live cells is plotted against time, we have distinct phases that we can see in this curve. So we're going to take a closer look at this growth curve and understand the different phases. So here's our illustration of this, and I'm going to come back to this, but when we're talking about each of the sections, you can think of this chart here, this graph. So on this side we have time, this is our x-axis, we have time, so as we're going along in time, and then on this side we have our logarithm of living bacterial cells, which basically is the number of cells. So going this direction would be an increase in number of cells, and going this direction is over time. And then we're going to talk about each of these different phases. So we're going to start with number 1 here, the lag phase. So the lag phase is our beginning of our growth curve, and that represents a small number of cells. So we start out with a small number of cells, and this is referred to as the inoculum. So the inoculum are the cells that we've added to a fresh culture medium. So if we take a broth tube, for example, which is a liquid agar or a liquid broth, we're going to take a small number of cells, let's say on our inoculating loop, we'll pick up some cells from an agar plate, and we will inoculate that broth tube. So we're going to take just a dab of cells and swish it around in our broth tube. That's our inoculum, and we are adding that to our culture medium. So for example, our nutritional broth. And then that's going to support growth, because in our nutritional broth we have the nutrients that are required for that particular organism to grow. So the initial phase of the growth curve is called the lag phase. So in this lag phase, this is when our cells are gearing up for the next phase of growth. So we already talked about binary fission. This is the stage, the lag phase, is when all of the cells are going through their normal everyday metabolizing, so living, so kind of like we do, taking in nutrients, processing those nutrients to build things within itself, and secreting its waste or getting rid of its waste. Also, during this time, it's gearing up for the next phase, meaning that it's going to be going through that process of making multiples of its different proteins, multiples of its enzymes, multiples of the different parts of the cell. So during this time, the number of cells does not change. So in the lag phase, we have the same number of cells that we stick in there, the inoculum, the number of cells we swish around when we put them in there with our inoculating loop. So the number of cells does not change in the lag phase. However, the cells are growing larger and are metabolically active. So again, this is the point where we're synthesizing our proteins that are needed to grow within this medium. So during this time, the cells are adjusting to being in a new medium. They're saying, okay, this medium is high in whatever, so we're going to make the proteins that we need, make the enzymes that we need, make the different things that we need in order to use this medium to grow in. So it's going to be synthesizing these proteins, making it adaptable to this particular medium. And then it's also going to be gearing up for going through binary fission, for making copies of itself. And so it'll be building up all of its other cell components so that it can go through binary fission. Also, if any cells were damaged, shocked during that transfer into the new medium, then that's when these cells will be going through repair. So in the lag phase, again, we don't have a growth of cells. They're going through repair. They're going to be making what they need in order to exist in the new medium. And then also they're going to be preparing for binary fission, so growing the cells. They're seen as being larger. So during the duration of the lag phase, or the duration of the lag phase, is determined by many factors. So we have our generalized growth curve, but the amount of time that an inoculum, or the amount of time that our culture is in lag phase, is going to depend on various things. So, for example, the species, the genetic makeup of the cells, the composition of the medium. So if we give them a very, very high in nutrient medium, or if we give them a medium that's not very high in nutrients, that's going to change the growth curve. And then also the size of the original inoculum. So if we give them a very small amount of medium, and we put a whole lot of inoculum in there, for example, there isn't going to be a lot of nutrients there. So we're going to see a different type of lag phase than if we were to put a very tiny amount of cells, say just a couple, in a huge amount of growth medium or a huge container of broth. Then they'll have lots and lots of nutrients. So we'll see different types of growth curves based on all of these different types of situations, all these different factors. So the lag phase is the first part of the phase, and we can see that on our graph here. So 1 is this lag phase, and again, if this line goes up, it would mean an increase in the number of cells of bacteria. Since it's not going up, remember, it's our lag phase, so we do not have an increase in the number of cells. That stays the same, and they're doing their regular stuff. And this goes on for a certain amount of time. So in our generalized graph here, the lag phase is from here until here. Okay, because during a lag phase, we don't have an increase in number of cells. Then the second part of our growth curve is the log phase, the logarithmic growth phase, or the log phase. So it's also sometimes called the exponential growth phase. And this is when the cells are actively dividing by binary fission, and then their numbers are going to be increasing exponentially. So log growth, logarithmic growth, if you remember back to some math classes, that's exponential. So it's not just 1 going to 2, going to 3, going to 4. If we backed up a little bit, there's actually an image that showed our one single cell through binary fission is going to become 2 cells, but then those 2 cells aren't just going to go to 3 cells, which would be linear growth. Instead, we see logarithmic growth, which means that those 2 cells are actually going to become then 4 cells, and then those 4 will be 8, and on and on. So instead of seeing linear growth, we see exponential growth or logarithmic growth. So for any given bacterial species, the generation time under specific growth conditions, so whether, what kind of nutrients are available, how many of them or how much of them, the temperature, if it's optimal, if it's not, pH, if it's optimal or not, etc., is genetically determined. So depending on the species, depending on the genetics for a given bacterial species, under certain growth conditions, we can know their generation time. This generation time here is called the intrinsic growth rate. So we will know the growth rate given certain parameters. So there's lots of information that's already out there through many, many years of research on very specific organisms. For example, E. coli, given a certain amount of nutrients, given a certain temperature, given a certain pH, then we'll know exactly how much E. coli will have at a certain time, because we have grown this and done this over and over again. We know their growth curve. So during the log phase, the relationship between time and the number of cells is not linear, as I mentioned, but it's exponential. So the growth curve is often plotted on a semi-logarithmic graph, as we see in the graph up above, which makes it look like it's linear. Meaning, so on our graph, if we have our time here and our numbers here, so like 1, 2, 3, just for simplification, when we see something moving this direction, that is our linear. It's a linear relationship. When we move up 1 unit here, we're moving up 1 unit here. If we move up 2 units here, we move up 2 units here. So that would be a linear relationship. On our graphs that we see for our growth, it does look to be linear, but that's just simplification. So here, to show you on this example here, we've ended our lag phase rather, starting our log phase, and we know it's the log phase because we see this increase in growth, an increase in number of cells. And again, it looks very linear, but it indeed is not linear, and it is exponential. It's logarithmic, which is why it's called the log phase. So we have an exponential increase in the number of living bacterial cells. So we see this huge spike here up to this point. So from this point to that point, we have our log phase. So here's actually an image, or some graphs here to show the difference between in A, when it's plotted on an arithmetic scale, the growth rate actually resembles a curve here, versus B, when plotted on a semi-logarithmic scale, then it appears linear. So we actually see linear growth, even though, as I mentioned here, this is our exponential growth. So when we go from one generation to the next, here we've doubled in size, and then same thing here. When we go from this generation to the next, we've doubled in size. It's not 1 to 1. If it was linear, we would go from 1 to 2, and then from 2 to 3 would be linear, and that is not what we see in bacterial growth. Again, it's exponential. So then the cells in our log phase show constant growth rate, so they're just exponentially growing over and over again, and uniform metabolic activity. So their metabolic activity is uniform in that they are using all the nutrients they can at their disposal to be creating all the proteins needed, all the enzymes needed, going through and doing that DNA replication, going through binary fission, and then just doing that over again. So for this reason, cells in the log phase are preferentially used for industrial applications and research work. Meaning, what we try to do in the laboratory and what people try to do in different industrial applications, so in different places across the world, is they are trying to use cells in the log phase. That's kind of the idea, is that when cells are in their best, or at their best, they're going through, they have great metabolic activity, they're happy, they're making products, they're using products, they're going through this process of binary fission. That's when we want to use these things, because they're going through their metabolic activity. And in industrial applications, for example, that's when we're going to be getting the products from them. So if a bacteria is used in industry, likely the bacteria is creating a product, and we want it to be in the log phase in order to get that product, because that's when it's doing the best job of making that product. Also in research work, we want them to be in their best at their best point, because we want to see them function at their optimum. So the log phase is also the stage where bacteria are the most susceptible to the action of disinfectants and common antibiotics that affect protein, DNA, and cell wall synthesis. So since these bacteria are happy and they're grabbing things from their exterior and they're bringing them into their cells, that's when we have the best chance at killing them through disinfectants and antibiotics, because they're going through this process of building these things. And a lot of antibiotics work by disrupting that. So if they're trying to make proteins and we have an antibiotic that disrupts their protein making, their protein synthesis, then that's going to go through in that process. Rather than if they're not going through protein synthesis, then that antibiotic may not work. So after our log phase, then we have what's called the stationary phase. So this is when the number of cells, or as the number of cells increases through the log phase, we have several factors that contribute to a slowing of the growth rate, and that's what we see in the stationary phase, as indicated by the term stationary. It's stable, it's not really going anywhere. So we have our waste products are starting to accumulate. So if we have our plate, remember we're in a closed culture, we're not letting anything in or out. So we have our waste products are starting to accumulate on the plate or in the broth, and the nutrients are getting used up. So we're running out of nutrients, we're creating lots of waste. So at this point, we're not going to be in the log phase anymore, because the bacteria aren't as happy. They don't have all of these nutrients around in a relatively clean space. Now they're running out of nutrients, and now they're kind of swimming around in their own waste products, which is less than ideal. In addition, a gradual depletion of oxygen begins to limit aerobic cell growth. So if we have our tubes and we have our lids on our tubes, we're going to be using up, or the bacteria will be using up that oxygen, so they're going to be running out of oxygen, which would be like a nutrient, running out of what they need in order to grow. So this combination of unfavorable condition is going to slow and then finally stall the population growth. So in the stationary phase, we see this slowing and then stalling. So the total number of live cells reaches a plateau, and this plateau is called the stationary phase. In this phase, the number of new cells created by cell division is now equal to the number of cells that are dying. So we have a lot of cells that are dying because they're running out of nutrients or they're being affected by the increase in waste products. And at the same time, we still have some cells that are going through binary fission because they're finishing up their process of binary fission, or they happen to be in an area of the broth or the plate that has some nutrients still. So they're able to go through binary fission. So we have this this equaling out here. So we have some cells that are going through binary fission, cell division, and then we have some cells that are dying. So the total population of living cells is relatively stagnant in the stationary phase, so it's stable. The culture density in a stationary culture is constant. So we have this constant culture density, meaning the number of cells stays the same, because we have this equal number of making and dying. So the culture's carrying capacity, or maximum culture density, depends on the types of microorganisms in the culture and the specific conditions of the culture. However, the carrying capacity is constant for a given organism grown under the same conditions. So what we see is, again, we know these things for certain organisms. We've studied them. We know that once they get to a certain point, they've used up their nutrients. They've got a lot of waste. This varies, depends on the types of organisms in the cell culture and the conditions, but We have our carrying capacity, the number of cells there that it can hold, so the number of cells that certain medium can hold given the specific conditions, and that's constant. So we know that number for various organisms or many organisms, and we reproduce that. So if we grow it under the same conditions over and over again, we'll get those same numbers. During the stationary phase, cells switch to a survival mode of metabolism. So rather than being in the log phase, they're just swimming in it, they're all happy, they're using up nutrients, they're reproducing. In the case of the stationary phase, they get into this survival mode. They say, okay, now we're running out of nutrients, we got a lot of waste around, so our growth is going to slow. Also does the synthesis of peptidoglycans, proteins, and nucleic acids. So when the cells are like, hey, wait a minute, there aren't a lot of nutrients, I'm going to go ahead and slow things down here. I'm going to slow down making my peptidoglycans, my proteins, nucleic acids, because it's not looking really likely that I'm going to be going through binary fission. Thus, our stationary cultures are less susceptible to antibiotics that disrupt these processes. So unlike the log phase where they were going through these processes very fast because they had lots of nutrients, we could disrupt that. We could go in and we could cut that short and kill the bacteria. In this case, since they're going so slowly, they've really slowed down their use and their synthesis. Then if we try to apply antibiotics at this point, then what we see is they're less susceptible to those antibiotics because they're not going through these processes, and if these antibiotics interrupt these processes and they're not doing it, then we're not going to be killing the cells. In bacteria that are capable of producing endospores, many cells are going to undergo sporulation during this stationary phase. So this is when we start to see sporulation because they're running out of nutrients. Secondary metabolites, including antibiotics, are synthesized in the stationary phase. So depending on the microorganism, this is when we start to see things like endospores forming, but then also we see things like secondary metabolites. So at this point, what we see in certain bacteria or certain pathogens, and it even says it here, in certain pathogenic bacteria, the stationary phase is associated with an expression of virulence factors. So products that contribute to a microbe's ability to survive, reproduce, and cause disease in a host organism. So at this point in the stationary phase, some of the cells, some of the culture, depending on the species of course, is going to go through the process of making endospores, kind of close up shop and wait for times to be better. Others are going to be producing secondary metabolites. So they're going to be making things like toxins and antibiotics that they're going to secrete to try to kill nearby bacteria, so other types of species, in order to get those out of the way so that they can use the nutrients that are available. So kind of a survival of the fittest here. So they're going to be trying to survive on their own, and they're going to be producing these secondary metabolites to try to get rid of anything else that might take up what it needs to survive. And then also these virulence factors. So we see certain pathogenic bacteria that are going to be undergoing or making toxins and things that will be harmful to the host. So not only just other bacteria that will be making things that are harmful to other bacteria in the area. For example here, quorum sensing. So quorum sensing is sensing the amount of things around it. So a quorum. So sensing its area, what's around it. Staphylococcus aureus initiates the productions of enzymes that can break down human tissue and cellular debris. And so this organism, the Staph aureus, is going to sense that there aren't a lot of nutrients. It's going to sense that there are a lot of cells around it. For example, human cells are actual body cells inside of our body. And it's going to start making enzymes that are going to kill those cells so that the bacteria can spread to new tissue where there are more nutrients. So it's going to send out these enzymes, kill off the stuff that's around it so it can get to a space where there's more nutrients. So if we look at our growth curve, again, if we go up here, we have now talked about our lag phase, which is when we have no increase in the number of cells at the beginning here. It's kind of getting situated to its environment. Then we have the log phase, which is where we have exponential growth from the end of the lag phase up here to where it stops, going up exponentially. And then we just talked about the stationary phase. So this is where we have our plateau. And so remember, the rate of cell division and death are about equal. So the same number of cells that are being made are dying. And so then we have this stationary phase right here, again, where we see endospores being made, where we see cells trying to survive, send out toxins and other metabolites. And then now we're going to talk about the fourth stage here, which is death or the decline phase. So our death phase, as it is called, is when our culture medium is going to be accumulating toxic waste and nutrients are going to be exhausted. So we've gone through a stationary phase where everything's slowed down, they're still using nutrients in order to survive, and then we get to a point where all the nutrients are used up and there's so much waste, so many waste products that the cells are now dying. So they're going to die in greater and greater numbers. Soon, the number of dying cells is going to exceed the number of dividing cells. So we get to a point where that's no longer equal in the stationary phase, and now there are more cells dying than are being dividing. So leading to an exponential decrease in the number of cells. Aptly named the death phase, also called the decline phase. So we see a decline in the population. Many cells lyse and release nutrients into the medium, allowing survival cells to maintain viability and form endospores. So what we also see in this death phase is exactly that. We see these cells that are like, okay, I can't handle them, that's it, I'm done. They pop, they lyse, and they release all of their stuff into the medium. Then the cells that are around it can pick up their stuff and survive long enough to either get to a place where there are more nutrients so that it can go through division, or it can form endospores using those nutrients. There are a few cells, the persisters, those are those that persist through, and they are characterized by slow metabolic rate. These persister cells are medically important because they are associated with certain chronic infections. So those are the ones that kind of hang out even though the nutrients are not really great, there's a lot of toxic waste around, they're going to persist. And when they persist, then we get these chronic infections in humans like tuberculosis. And then these don't respond to antibiotic treatment because they're hanging out, but they're not metabolic at that point. They're not making the proteins. They're not going through the process of bringing in nutrients. They're just kind of trying to hang in there until, again, the nutrients are good, and then they can go through their binary fission. All right, so let's go back up and just do our general overview of our graph here. So remember, this is our growth curve. When we're looking at a culture, a closed culture, we see our lag phase here. When we put our inoculum in new medium, it's getting used to it. We don't see an increase of number of living cells. Then we have our log phase, where we have an exponential increase going up here. Again, it's not linear, but it's exponential, or logarithmic, why it's called the log phase. Then we have our stationary phase right here, where we have this plateau. We have the same number of cells dividing as we do dying. And then the phase we just talked about, which is the death phase or the decline phase. So we have our exponential decrease in the number of cells. So we just see this go all the way down until eventually we hit zero and the entire culture is dead because there aren't any more nutrients for it, and there's such a buildup of toxic waste products that the cells can no longer live. That said, again, we may have some that have produced endospores in this stationary phase, so we may still end up somewhere in here with our endospores that are able to continue on, and also our persisters, which will then continue on. So we may not have a complete death, but get to the point where we just have those endospores hanging out, which are not living or vegetative cells, but then can become, again, later when they're in an ideal environment. But then also we have those persisters that will keep floating around until they do get somewhere and then can contribute to that chronic infection. So then when we get to our sustaining of microbial growth, we took a look at our growth pattern, and if we want to sustain microbial growth, again, we had mentioned that in industry, for example, it is useful to have the cells be in the log phase, the logarithmic phase, where they're metabolically active and they're producing whatever it is that that industry is producing using those microorganisms. What we can see here is that if we want to keep them in this phase, we can use something called a chemostat. And this is an image of a simplified chemostat here. So we have our growth pattern that we saw, our growth curve, and that takes place in a closed environment. So remember that nutrients were not added and the waste and dead cells were not taken away. And then we saw that regular growth curve, which had our log phase, which is the ideal phase for our purposes, but then it hits that stationary phase and then the death phase. It is advantageous, however, to keep them in the log phase for industry. So for example, industries that harvest microbial products, so they're going to use this chemostat, and it's used to maintain a continuous culture in which nutrients are supplied at a steady rate. So you can see here that we have this container here that in the blue is where we have our bacterial culture, and we can add feed, so there's some sort of intake or some sort of tube, for example, that's going to be bringing nutrients into the bacterial culture. We have this kind of stirring up that's occurring, not only to make sure that the nutrients get throughout the culture, but also they show bubbles here to show that it's a suspension or a mass of cells that needs oxygen, for example. It's an aerobic bacterium, so they're going to be adding oxygen in this way by stirring up the mixture, adding bubbles, bringing the air into it. So we're adding nutrients to one side, we're mixing it up, bringing the nutrients throughout the mixture, getting oxygen in there, and then we have on the other side, we have our effluent, which is where we can take stuff out of this chemostat. So they'll be pulling things out so that they can use them to take those metabolic products, microbial products, and then getting rid of the dead cells and getting rid of some of the waste in the process. So we have our controlled amount of air is mixed in for aerobic processes, we have our nutrients getting in, and then at the same time we have our bacterial suspension is removed at the same rate that we add our nutrients in, so that we maintain this optimal growth environment, so that we maintain this logarithmic rate. So we're constantly on the log part of the scale, and we're not ever reaching that stationary phase or the death phase.